What if your medicine could do more than just act? It could think, sense, and respond like a living system. Synthetic biology is making this idea possible by transforming microbes into programmable living therapeutics. But what exactly are living therapeutics? Living therapeutics are an emerging class of medicines made from genetically engineered microbes. These microbes are programmed to carry out specific biological functions inside the body, such as detecting disease-related biomarkers, synthesizing therapeutic compounds, or suppressing harmful pathogens. Unlike traditional drugs that often affect the whole body, living therapeutics are designed to operate with spatial and temporal precision, offering a more controlled, adaptive, and targeted form of treatment. Living therapeutics function much like programmable biological computers. They rely on an integrated system of components that allow them to work as effective and safe therapies. These key components include sensors that detect biological signals, logic circuits that process information and determine when to respond, effectors that produce and release therapeutic molecules, and kill switches that provide built-in safety control. Let's take a closer look at how each of these core components work together to enable a precisely controlled and responsive therapeutic system. First, the engineered microbes monitor their environment using synthetic molecular sensors, which are specifically designed proteins or genetic elements that can detect biological signals such as inflammation, bile acid, or changes in temperature. When a relevant signal is identified, it's transmitted inside the microbe to initiate a response cascade. This signal then reaches the next component of living therapeutics that we'll be discussing, the microbe's genetic decision-making systems, otherwise known as logic circuits. Logic circuits function like molecular decision makers, applying simple rules that are similar to digital electronics. The rules may be applied through logic gates, which will stop the signals from being transmitted if the correct conditions are not met. For example, an AND logic gate might require two separate signals like inflammation and a changing temperature to be present before activating a therapeutic gene. This step ensures that the microbe responds only when the conditions are just right, making its actions more selective and safe. When the logic circuit determines that all necessary conditions are met, the microbe initiates the expression of specific therapeutic genes. These genes encode effector molecules such as anti-inflammatory cytokines, antimicrobial peptides, or enzymes that target disease processes. As a result, the engineered microbe acts like a microscopic drug factory, delivering a targeted treatment directly at the site of the disease. But how do we ensure that engineered microbes don't escape their intended environment or act unpredictably within their body? To minimize risks and maintain control, synthetic biologists incorporate kill switches, engineered genetic circuits that trigger cell death or deactivate microbial function under predefined conditions. These can include environmental cues such as exiting the gut or the absence of an essential synthetic nutrient. With this full system in place, sensing, decision-making, therapeutic action, and self-limiting control, we can now look at how these components are already being applied in real-world medical settings. Living therapeutics are already being explored across a wide range of medical conditions, with engineered microbes being tailored to detect disease-specific signals and release treatments with precision. For instance, in metabolic disorders like phenylketonuria, microbes can be designed to degrade excess phenylalanine. Furthermore, in certain cancers, tumor-targeting strains are being developed to deliver immune-modulating agents directly within the tumors. These organisms are also being formulated for different delivery routes, which play a key role in the success of a living therapeutic. To function effectively, engineered microbes must reach the right tissue and remain active only when needed. For gut disorders, this might mean formulating the microbe to survive stomach acid and activate in the intestine. For skin applications, microbes are applied topically and engineered to detect wound signals. And as mentioned earlier, in some cancers, therapies can be injected directly into tumors. By aligning the delivery route with biological context, synthetic biologists help ensure precision, safety, and efficacy in treatment. Now let's take a closer look at how this technology is being applied in one of its most advanced areas, treating gut inflammation. The gut is one of the most promising and well understood environments for living therapeutics because it's easily accessible and home to many types of microbes. A well-known example of this involves modifying two probiotic strains, E. coli Nissl 1917 and Lactococcus lactis, to act as inflammation sensors. These engineered microbes are designed to detect signs of inflammation in the gut, like high levels of nitric oxide or reactive oxygen species, which are common molecules during conditions like inflammatory bowel disease. When the microbes sense these signals, their built-in genetic circuits trigger the production of interleukin-10, a substance known to reduce inflammation. Interleukin-10 is released directly into the affected area of the gut, helping to calm the immune system and promote healing. Because the treatment is delivered only where it's needed, it can reduce side effects compared to drugs that affect the whole body. So to sum it up, living therapeutics are paving the way for a new generation of precision medicine. Fueled by advances in biosensing, programmable logic circuits, and targeted delivery systems, these engineered microbes are becoming powerful tools for treating complex diseases with site-specific precision. As this field matures, researchers are beginning to explore a broader set of possibilities for the next generation of applications. In the future, living therapeutics could be used not just for treatment, but for prevention, tailored to individual microbiomes, or even designed to act as internal diagnostics that monitor and report on tissue health in real time. As we continue to unlock the full potential of synthetic biology, living therapeutics may soon become a cornerstone of personalized, responsive, and safer medicine. Thanks for watching.